Hello everyone, welcome back to Meds.Concepts. Today I'll be introducing gram staining, which is one of the most fundamental and widely used techniques in microbiology. Gram staining is a differential staining technique that distinguishes bacteria based on how they respond to a standardized sequence of dyes. In simple terms, it helps us classify bacteria into two major groups, gram-positive and gram-negative. This technique is extremely important because it provides rapid information about bacterial morphology and classification, even before culture or molecular results are available. It allows clinicians and microbiologists to make early decisions in diagnosis and treatment. The key concept behind differential staining is selective retention. Some cells retain the primary stain, while others lose it and take up a counterstain. This creates two distinct color outcomes, making it easier to differentiate between bacterial types. Using this method, bacteria are broadly grouped into gram-positive and gram-negative categories. This initial classification is essential for further identification and understanding of bacterial behavior. On the right side, you can see a step-by-step visualization of the gram staining process. Let's now move forward to understand its principle in detail. The main principle of gram staining is that the structure of the bacterial cell wall determines whether the stain is retained or removed. Gram-positive bacteria have a very thick peptidoglycan layer, approximately 90%. When crystal violet and iodine are added, they form a crystal violet iodine complex. This complex gets trapped within the thick cell wall. As a result, even after decolorization, gram-positive bacteria remain purple. In contrast, gram-negative bacteria have a thin peptidoglycan layer and an outer membrane. During the decolorization step, alcohol dissolves the outer membrane and removes the crystal violet iodine complex. As a result, these cells become colorless and later take up the counterstain, safranine, appearing pink or red. Gear steps of gram staining. Now, let's understand how the process works. It involves four main steps. First, we add crystal violet, the primary stain, which stains all cells purple. Second, we add iodine, which acts as a mordant. It combines with crystal violet to form a large complex that gets trapped inside the cell wall. Third, we apply the decolorizer, usually alcohol or acetone. This is the most critical step. It removes the stain from gram-negative cells while gram-positive cells retain it. Finally, we add safranine, the counterstain. This stains the previously colorless gram-negative cells pink. Microscope final observation. After staining, when observed under the microscope, purple-colored cells are gram-positive bacteria as they retain the stain due to their thick cell wall. Pink or red-colored cells are gram-negative bacteria. This distinction is very important because it helps guide appropriate antibiotic selection. While performing gram staining, certain errors can lead to incorrect results. Over-decolorization. In this case, excessive alcohol removes the stain even from gram-positive cells. This leads to false negative results, where gram-positive bacteria appear pink. Under-decolorization. Here, insufficient alcohol fails to remove the stain from gram-negative cells. As a result, gram-negative bacteria may appear purple, leading to false positive interpretation. Thick smear. If the smear is too thick, the stain cannot penetrate evenly. This obscures cell morphology and can lead to incorrect interpretation. Old culture. In older cultures, especially gram-positive bacteria, the cell wall may become damaged. This can result in gram-variable or false gram-negative results. Hospital applications in medicine. So, where is gram staining actually used? It is not limited to the laboratory. In hospitals, it is widely used for rapid bedside diagnosis, identifying the type of infection, and initiating appropriate antibiotic therapy. It also provides information about bacterial shape and arrangement, such as chains or clusters. To conclude, gram staining remains a timeless and essential tool in routine microbiology. High-voltage, rapid method. One of its greatest advantages is speed. Gram staining provides actionable information within minutes, allowing clinicians to make early and informed decisions. Money bag, low cost. It is also highly cost-effective. It requires only basic reagents and minimal equipment, making it accessible in a wide range of laboratory settings. Microscope diagnostic value. Gram staining offers immediate insight into the type of organism involved. It helps in identifying whether the bacteria are gram-positive or gram-negative and gives clues about their potential pathogenicity. Brain foundational skill. Beyond diagnosis, it is a fundamental laboratory skill. It serves as the foundation for more advanced microbiological techniques and interpretation. Thank you. If you found this explanation helpful, don't forget to subscribe to Meds.Concepts. Press the bell icon so you never miss an update and share this with your classmates. Let us know in the comments which topic you would like to see next. Keep learning and see you in the next video.